Greetings to everyone, to our dear panelists and researchers. I am Princess Angelica Besonia, as part of Team CICID representing the Angeles University Foundation Integrated School, along with my esteemed co-researchers Maria Angelica Gomez, Jules Hyacinth Macasaet, and Nika Banyes. We are thrilled to present our research titled Biodegradative Activities of Pleurotus austriatus and Volvariella bulbacea on high-density polyethylene and oxo-biodegradable plastics. This study focuses on aerobic biodegradation, which breaks down organic chemicals, releases CO2 and water, and investigates selective etching as a method to catalyze biodegradation and modify surface properties. Additionally, the researchers investigate the potential of lignocellulosic fungi to degrade plastic waste by utilizing lignin, cellulose, and hemicellulose, proposing that enhancing fungal growth through baking and etching processes could improve the degradation of various types of plastic, highlighting anaerobic biodegradation as a promising solution for addressing plastic waste. Moving forward to the methodology. For the collection, the researchers obtained laboratory-grown peer cultures of Pleurotus austriatus and Volvariella bulbacea, while collecting HDPE plastics from their households and purchasing oxo-biodegradable plastic, which were then washed and cut into 1.5 by 1.5 centimeter strips. For the media preparation, potato dextrose agar was used as a carbon source for the fungi. The HDPE and oxo-biodegradable plastics, on the other hand, were subjected to three different treatments: baked, etched, and a combination of both. The baking process involved preheating a laboratory oven to 120 degrees Celsius for 4 minutes. For etching, a mixture of ferric chloride and citric acid solutions was prepared, and the plastics were soaked in this solution for 10 minutes. The control group, on the other hand, did not undergo any treatment. Finally, for the experimentation setup, the experiment involved two groups, a control group and an experimental group, each with three replicates. The groups were divided based on the type of fungi and the treatment of the plastics.
Moving on to the results and discussion. The first experimental setup shows the micrographs of Volvariella bulbacea and oxo-biodegradable plastic. This showed high biodegradation after baking and etching with lignocellulosic enzymes and fungal colonization. The oxo-biodegradable film showed a rocky mass indicating fungal colonization, as also observed in the study of Promilla endomesh, where LDPE was tested in Aspergillus species. Groups tested with etching manifested average plastic degradation. For the second experimental setup, this shows the micrographs of the same species, this time tested with HDPE plastic. This exhibited slight degradation on baked plastics as there was only minimal disruption on the plastic surface. The group that underwent etching process displayed high degradation. For the third experimental setup, this shows the micrographs of Pleurotus austriatus tested in oxo-biodegradable plastic, and it showed the highest biodegradation among the group pre-treated with etching. A huge lump can be observed on the plastic surface as it projects diggings surrounding the lump. Lastly, for the fourth experimental setup, this time, Pleurotus austriatus is tested on HDPE plastic, and it demonstrated the highest degradation on baking and etching plastic. In conclusion, the SAM images of the plastic sample showed varying morphological effects of a tooth fungi on the surface of HDPE and oxo-biodegradable plastics. The control groups demonstrated no disruption in the plastic surface. Pleurotus austriatus displayed high degradation on baked and etched HDPE and etched oxo-biodegradable plastics. Volvariella bulbacea showed the most significant degradation on etched HDPE plastics. This study was funded by the Third Young Innovators Program of the UST Bishop. The team would also like to express their sincere thanks and gratitude to Angeles University Foundation Integrated School for providing support for the betterment of the study. The references used in the study are as follows. Once again, this has been Team CICID. I am your presenting author, Maria Angelica D. Gomez, along with Princess Angelica Bisogna. Thank you for listening.